Hi, this presentation is on pharmacology is simplified overview. Pharmacology is science which deals with study of drugs. And drugs are substances, either chemical or biological, administered or applied to body to produce beneficial effects in form of either curing diseases or providing relief from symptoms, or drug may be used as a diagnostic agents. Sources of drug, natural sources like from plant, example quinine from cinchona bark, animal source, insulin, microbial source, penicillin, semi-synthetic. These are synthesized from naturally occurring product like amoxicillin is produced from penicillin in lab. That's why it is semi-synthetic. Chemical synthesis, example ciprofloxacin, biotechnology source, example insulin. Pharmacology having two parts, pharmacodynamics and pharmacokinetics. Pharmacodynamics is what drug does to the body, or what are the effects of the drug in the body along with its mechanism of action. Pharmacokinetics is what body does to the drug. So or it's also called study of EDME, study of absorption, distribution, metabolism and excretion. Pharmacodynamics, what drug does to the body, it involves mechanism of action and important physiological and biological effects. Mechanism of action, example, for example, adrenaline increases heart rate and force of contraction by activating or stimulating beta 1 receptors. Mechanism of action, drug can act through various mechanisms. First, it's physical. Some drugs used in constipation, they act by increasing the bulk. Example is psyllium. The psyllium absorb water and swell up in the gastrointestinal tract and increase the bulk and put pressure in GI tract. So help in constipation. Action through chemical action. Antacids neutralizes gastric acid in stomach used in acidity problem. Action through enzymes. Example captopril, which is antihypertensive, acts by inhibiting angiotensin converting enzyme or ACE. Action through ion channels. Example, amlodipine acts by blocking calcium channels. Action through transport proteins. Example, omeprazole acts by inhibiting proton pump, which is a protein. Action through receptors, and a drug on receptor binds to receptor, and on receptor drug can either act as an agonist or antagonist or partial agonist. Agonist binds to receptors, which is called affinity, and produces effect, which is called efficacy. Example, adrenaline binds to beta 1 receptors in heart and increases heart rate and force of contraction. Antagonist Antagonist binds to receptors and prevents binding of agonist on receptors. So antagonist have affinity but no effect or efficacy of its own. Example, propranolol binds to beta receptors in heart and prevents effect of adrenaline. Prevents binding of adrenaline on beta 1 receptors. Partial agonists, which partially stimulates receptor and have partial effects. Type of receptors. Basically, four types of receptors: ligand gated ion channel, G protein coupled receptors, kinase linked and related receptors, and nuclear receptor. Ligand gated ion channels receptors, they are membrane receptors. Example, acetylcholine nicotinic receptors, GABA A receptors, glutamate receptors of NMDA, AMP, and kinase types. Second type is G protein coupled receptor. There are also membrane receptors, and they couple to intracellular effector system via G protein. There are These are receptors for many hormones and transmitters. For example, acetylcholine muscarinic receptors, adrenergic receptors, and chemokine receptors. Third type of receptors are kinase linked and related receptors. These are also membrane receptors. That single transmembrane helix linking extracellular receptor domain to intracellular kinase domain or binding site. Insulin example is insulin receptors, growth factor receptors, cytokine receptors, and arterial natriuretic factor receptors, nuclear receptors. They are intracellular receptors. They regulate gene transcription. They are monomeric. They have monomeric structure with separate receptor binding and DNA binding domains. Example receptors for steroids, thyroid hormone, retinoic acid, and vitamin D. Routes of drug administration, local and systemic. Local may include topical cream, lotion, and ointments for local action on skin. Eye drops, ear drops, nasal drops for local action or effect in eyes, ears, and nose. Systemic route of administration includes oral or enteral. Like example, like taking tablet, capsule, or suspension or syrup via mouth. Second systemic route is parenteral or injection, which may be intravenous, subcutaneous, intramuscular, or intrathecal, etc. Third systemic route is sublingual, in which the tablet or drug is placed under tongue and from where it is absorbed. Rectal route, drug is administered through rectum, either in the form of suppositories or enemas. Next systemic route is cutaneous or transdermal routes, which is not for local action but for absorption of drug in the blood, like nicotine transdermal patches, which are applied on the skin, and from the skin they are absorbed in the blood for systemic action. Inhalation route, example different inhalers used in asthmatic patients, and there are also these asthmatic patients, these inhalers are for local action on in the lungs and bronchi. But nowadays there are other inhalers are also available which are for systemic action, in which the drug is administered through inhalers and they are absorbed from the lungs and drug goes to the systemic circulation for systemic action. Similarly, nasal route of administration is also used for systemic action. These nasal drops, they are absorbed from the nasal cavity and drug goes to the circulation. And this is for systemic effect, not for local effect. Pharmacokinetics is study of ADME or study of absorption, distribution, metabolism or excretion. So pharmacokinetics is how body deals with the drug. So body will absorb the drug from site of administration. Body will distribute the drug within the body. Body will metabolize the drug in the body by various organs like liver. Body will finally excrete the drug by various body systems like urinary system. Absorption of drug, which is transport of drug from administrative site across cell membrane. Absorption may occur by diffusion, in which lipid soluble drug diffuses across the membrane, or by filtration through aqueous protein pores, like water soluble drug, they pass through the pores. Third is carrier mediated active transport, which involves the use of a carrier protein. And fourth is pinocytosis, in which the membrane, the cell membrane, they engulf the drug molecule on one side of membrane and they will leave the drug on other side of the membrane. Like insulin while crossing blood-brain barrier use this pinocytosis. Factors affecting absorption like aqueous and lipid solubility. Lipid soluble drugs are having more absorption or faster absorption. Second factor is concentration of drug at the site of administration. Third factor is area of absorption surface, like many drugs absorb from the intestine, which is having larger surface area, and not from the stomach, which is having smaller surface area. The next factor is vascularity of absorption site or blood supply at the site of absorption. And next factor is route of administration, like parenteral or the intramuscular drug administered drug are absorbed faster than the oral drug. Oral absorption, which is diffusion of drug through GI epithelium into portal circulation. Oral absorption depends on aqueous and lipid solubility of the drug, particle size or formulation type, like rapid absorption from solution formulation compared to tablet or capsule formulation. Next factor is GI motility or gastrointestinal motility, like diarrhea and some drugs they affect GI motility, and because of that they affect the drug absorption. Like in diarrhea, many drugs are not properly absorbed, and drug which increase the GI motility they will decrease the absorption of certain drugs. Next factor is splanchnic blood flow or blood flow to the liver. Food intake increases the blood flow to liver, and example is propanol absorption is increases when it is taken after meal, because meal increases the blood flow to liver, so more drug will be absorbed. Next factor is psychochemical factors, which include some drug interaction, like calcium rich food binds to tetracycline. So calcium binds to tetracycline and prevents its absorption. Distribution of drug to all parts of body by plasma. Distribution depends on plasma protein binding. So binding of drug to the protein which are there in the plasma is called plasma protein binding. Next factor in distribution is molecular size of the drug. Next factor is degree of ionization of drug, lipid solubility of drug, and tissue protein binding of the drug. The binding of drug to the proteins in the tissue is called tissue protein binding. Distribution to special tissues, like distribution to CNS happens through blood-brain barrier, which does not allow all the drugs to pass to the CNS. Distribution during pregnancy across the placenta. Placenta is the barrier between the baby and the mother, so all drugs they do not pass through the placenta. The secretion of drug in the milk. Metabolism is biotransformation of drug to active or inactive metabolite by special enzyme system, for example P450, cytochrome P450 enzyme system. Metabolism mainly happens in liver, which is most important metabolizing organ, but metabolism can take place in all tissues except hairs and nails. There are two phases of metabolism, phase 1 and phase 2. In phase 1, hydrolysis, oxidation and reduction occurs, and often this phase introduces a reactive group. For example, it adds hydroxyl group to the drug, and it takes place mainly in the liver, and often it precedes phase 1 reaction. It involves many hepatic metabolizing enzymes, for example P450 enzyme system. Phase 2 metabolism system, in which synthetic reaction involving conjugation with glucuronide occurs. It takes place mainly in liver and usually leads to inactive or polar products that are readily excreted. So metabolism basically help in excretion of the product. It converts the drug or toxic substance into water soluble and polar product which can be easily excreted. First pass metabolism, drug absorbed from GI tract, they first goes to the liver. Many drugs are metabolized in the liver before they reach to general circulation, and that is called first pass metabolism or pre-systemic metabolism. And this first pass metabolism reduces drug viability even when the drug is well absorbed from GI tract. For such drugs, a much larger dose is needed when given orally as compared to when given parenterally. Because when given parenterally, the drug does not go to the liver first; it will go directly to the circulation. But when drug given by oral route, then from GI tract, it will first go to the liver, and there it is metabolized to some extent. So all the drug absorbed will not go to the circulation. Some will be metabolized through first pass metabolism, and there is a marked individual variation in first pass metabolism. Enterohepatic circulation. It happens when the drug from the liver is released in the bile, and from bile it goes to GI tract, and in GI tract it is reabsorbed and after reabsorption it again go to the liver, so the drug will keep circulating between GI tract and liver. That is called enterohepatic circulation. Excretion, most important route of excretion is renal, and renal route of excretion involves three processes: glomerular filtration, in which the drug will be filtered in the tubules; active tubular secretion, in which drug is directly secreted in the tubules from the blood; and third is tubular reabsorption. Most of almost 99% of the filtered volume of the blood which is taking place in the kidney, it is reabsorbed. Only 1% passes as the urine. So tubular reabsorption, which is passive diffusion across tubular epithelium. So whatever is reabsorbed, it will go again in the body and into the circulation. Extra renal route of excretion are hepatic. For example, secretion of drug in the bile, which goes to the GI tract, and from GI tract through feces it is excreted. Excretion through expired air, for example, respiratory system, the gases and ethanol, they are excreted through the respiratory system in expired air. Some drugs are also excreted through saliva, sweat, and milk. Interrelationship of absorption, distribution, binding, biotransformation, and excretion of a drug. So when you take a drug from site of absorption, it will go to the circulation or in the blood, and in the blood, drug may exist either as a free drug or bound drug. Bound drug will keep circulating in the blood; it will not go to the tissue. But free drug will go to the tissues. It will go to the site of action where it will produce its effect. It will go to the tissue reservoir where it will be stored, and the free drug will also go to the site of metabolism where it will be metabolized, and metabolites will again come back to the blood and they will be ultimately excreted out. Pharmacokinetic parameters: plasma half-life, or THA. It is the time required for plasma drug concentration or amount of drug in the body to decrease by 50%. Bioavailability is percentage of administered drug reaching circulation is bioavailability. Peak concentration. When maximum of peak concentration happens in the plasma after administration is called Cmax, and the time when this Cmax occurs is called Tmax. Time after administration when Cmax happens is called Tmax. Volume of distribution, or VD, volume of plasma that would contain total body content of drug at concentration equal to that in plasma. Renal clearance, CLR, volume of plasma containing amount of substance that is removed from body by kidneys in unit time is renal clearance. Dose. Dose is amount of drug given each time for full therapeutic benefit, and dose depends on age, body weight, or body surface area in case of certain drugs like anti-cancer drugs in some cases. The liver damage, the kidney damage, coexisting other diseases, they also affect the dose. And other medication used for certain diseases, they also affect the dose. Drug interactions. Two or more drugs given together can interfere with each other. Drug interaction may occur either before administration, for example, two injections when mixed together certain time, precipitations happen. That is interaction before administration. During absorption, one drug may decrease or affect the absorption of another drug. Interaction at protein binding site, one drug may increase or decrease the binding of other drug to the plasma proteins. Interaction at metabolism site, one drug may affect the metabolism of other drug. It may decrease or increase the metabolism of another drug. Excretion site interaction, one drug may decrease the excretion of another drug. For example, probenecid decreases the excretion of beta-lactam antibiotics, penicillin and cephalosporins. Drug interaction can lead to either potentiation or synergism of effect. When two drugs are given together, they may produce additive effect. They may also produce superadditive effect, in which effect of both drug in combination is more than effect of individual two drugs. So 1 plus 1 become 3, not 1 plus 1 is not equal to 2 in superadditive. It will become 1 plus 1 will become 3 or more than 2. Antagonism. Antagonism can be pharmacokinetic antagonism, in which one drug reduces the effectiveness of another drug by altering the pharmacokinetics or altering the absorption, distribution, metabolism, or elimination of another drug. Chemical antagonism. For example, chelating agent dimercaprol binds heavy metal and prevent their toxicity or effect. There can be physiological antagonism, in which two drugs producing opposite effects. For example, adrenaline increases heart rate and acetylcholine reduces heart rate. Tolerance. Tolerance is gradual decrease in response to a drug. Tolerance can be natural or acquired. Natural tolerance, some people are naturally tolerant. For example, beta blockers are less effective in some African black hypertensive people. Acquired tolerance, which develops with the use of drug. After some time of using the drug, the tolerance will happen. That is called acquired tolerance. Tachyphylaxis. Tachyphylaxis is a rapid development of tolerance. Drug effects on body. They can be therapeutic or desired effect, and these are called indications. But there can be side effects or adverse effects also. These are unwanted and undesirable effects. And with all the drug, you will see the precautions and warning. For example, precautions needed in dose selection in elderly people, in patients with liver disease or kidney disease. They will need lesser dose. There will be contraindication with drugs. Like drug is contraindicated if a patient is allergic to that drug. Adverse drug reaction. These embody a wide variety of toxic drug reaction. They may be dose related or non-dose related. They occur in therapeutic situations. Usual classification of adverse drug reactions are mild, moderate, severe, and lethal. Mild, they are mild in nature, may not require therapy, may not prolong hospitalization. Moderate, they require change in drug therapy and may prolong hospitalization or require special treatment. Severe adverse effects, potentially they are life threatening, may require they will require discontinuation of therapy, and specific treatment for severe adverse reactions are required. Lethal adverse reaction, directly or indirectly, they contribute to the death of the patient. Dose related and predictable drug reactions, these can be side effects, overdoses, toxicity. Side effects which are predictable, you can predict that because of pharmacological action of the drug, and they occur within therapeutic dose range. Overdoses, toxicity, they are predictable and they occur with overdoses or in excess doses. Non-dose related and unpredictable adverse drug reactions, these are either drug allergy or idiosyncratic reaction. Drug allergy, they are determined by antigen antibody interaction and they are independent of pharmacological properties. They are non-dose related because even small dose may produce drug allergy, and they are unpredictable. You do not know in which individual drug allergy can happen unless you test it. Idiosyncratic reactions are they are in unexpected and peculiar reaction. They occur in very small percentage of patients. They occur maybe some deficiency of some enzyme in the body system. Adverse drug reactions, different terms like teratogenicity, they are harmful effects of drug on fetus if taken during pregnancy. Mutagenicity and carcinogenicity, this is cancer producing ability of drug. Photosensitivity produces allergic or sensitivity reaction in presence of sunlight. Ectogenic disease or drug induced disease or physician induced disease. Diseases produced by use of drug, maybe because of various adverse or side effects of the drug. Like aspirin produces gastric bleeding. Coming to clinical trials, it is study of drug in humans to measure therapeutic efficacy and safety, and it requires approval of an independent ethical committee before start of clinical trial, and it requires very careful planning and execution, and clinical trials are very expensive. There are four phases of clinical trial. Phase 1 clinical trial, which is done in 20 to 80 healthy volunteers to determine safety and initial doses. Phase 2 clinical trials done in 100 to 300 patients to look for efficacy and side effects. Phase 3 clinical trials done in 1000 to 5000 patients for confirming efficacy and monitoring long-term safety. Phase 4 or post-marketing surveillance, which are done in large number of patients after marketing approval of drug for monitoring safety profile in large population, because in phase 1, 2 and 3 studies very small number of patients are used, but after approval the drug will be used in huge population. So there may be many undetectable side effects which are not detected in phase 1, 2 and 3 trials. So that's why the regulatory agency will ask for the post marketing surveillance on phase 4 study for monitoring safety profile in large population. Last slide, new drug discovery and development timelines. The journey of medicine starts with the discovery phase. Drug will be discovered, and after discovery, the development will start. The company will develop prototype formulation and will do the preclinical studies in lab animals and animal testing. And after preclinical study, IND will be submitted to the regulatory authority, and after IND approval, phase 1, phase 2 and phase 3 studies will be done. After completing phase 3 studies, the company will submit NDA, new drug application, with the regulatory agency, and then regulatory agency will review the application, and if it is approvable will approve the drug for marketing. And regulatory agency will usually ask for post marketing a phase 4 study to monitor the long term safety of drug in large population. And along with the drug discovery and development timeline, simultaneously the patent protection will also start. The After discovery of the drug, when you have the product or invention, the company will immediately file the patent application in home country, in foreign countries, and patent will be granted by patent office if it is grantable, and patent term will be 20 years from date of filing of patent application, which patent term can be extended. If there is a delay in regulatory approval, then through H-Vaccine extension in US, the patent term may be extended maximum to 5 years. Also in US, if there is a delay in grant of patent by patent office, again term can be extended by 5 years in US. And third process is the generic competition. So the branded drug or innovator drug, it is protected by patent for 20 years. So for 20 years, generic company cannot get approval for generic product, but they will start development of the generic product well before expiry of the patent. And they will, because generic development process will take time. They have to develop the formulation, they have to test the formulation, they have to do the bioequivalence viability studies, they have to compile the dossier. So the generic company will do the all development work and compile the dossier before expiry, and they can submit the dossier before expiry, but approval of the product, generic product, will happen only after expiry of patent. So they can file ANDA or abbreviated new drug application or generic application with FDA before expiry, but approval of generic drug will happen only after expiry of patent. So the FDA or regulatory agency may approve the generic drug if it is approvable after expiry of patent. And after approval, generic company can market the product. So that's all in this presentation. Thank you very much.